Hello, everyone, and welcome back to the Connect Science webinar brought to you by the Dana Farber Harvard Cancer Center and the Leukemia Lymphoma Society. As always, please feel free to post your questions using the Q&A tab. It's my distinct pleasure today to welcome Dr. Nick Haining, who was formerly our colleague as an associate professor of pediatrics at the Dana Farber Cancer Institute, Harvard Medical School, and an associate member at the Broad Institute, but about a year ago transitioned to a leadership position as vice president of discovery oncology and immunology at Merck Research Laboratories. Nick has made seminal discoveries about novel regulators of the immune response to cancer, and he's going to extend those findings in a talk for us today. So Nick, thanks so much.
Dave, thank you. And I thank you also for the invitation to speak and also for really organizing what I think is a great way for us all to stay connected. First, disclosures. I'm obviously an employee of Merck & Company. I also founded a company based on T-cell editing for CAR T-cell therapy. So this is what I'm really going to be talking about today. So, in essence, I think the developments over the last 10 years in CAR T-cell therapy and in checkpoint blockade have really taught us that the profound capability, the profound biology behind the therapeutic effects of T-cells acting directly or indirectly to achieve therapeutic effects in the tumor. However, I think it's clearly far too early for us to declare victory. I think there's still a great deal of work to do. So today what I want to do is start by talking about what I see as some of the challenges in the field of immuno-oncology, and what is some of the work that remains to be done, particularly around the ideas of trying to identify new targets that can build on the progress that we've seen so far, particularly with checkpoint blockade. And to do that, I want to talk about some of the work from my academic lab using CRISPR screens or genetic screens to identify regulators of the immune response to cancer that are present in tumor cells, and also to identify those that are present in T cells that might regulate the balance between an effector response and immune evasion. And although the work that I'm going to present today is from my academic lab, they clearly influence how I'm thinking, or how many of us are thinking in industry about how checkpoint blockade, how we can build on checkpoint blockade still further. And finally, if there's time, I just want to add a few perspectives about how this is my wife, Hayden, who's trying to very surreptitiously sneak behind the screen. And finally, if I finish, I just want to talk about a few perspectives. As Dave mentioned, I've recently moved from the position at the Dana Farber and over the lab at the Broad across the street, literally, to Merck Research Lab. So I thought it might be interesting to talk about some of the perspectives of that shift. So it's really these kinds of data that I think emphasize just how profound the shift has been in cancer care in the era of checkpoint blockade. This is obviously checkpoint molecule I'm particularly familiar with, pembrolizumab, and shows the monotherapy efficacy of this drug across a range of different cancer types. And the breadth of that efficacy across a diverse set of cellular and molecular phenotypes really underscores that checkpoint blockade has really become one of the small number of therapies, perhaps even limited only to radiotherapy, that has as broad a clinical scope of efficacy as it does. However, I think what's clear is that there's a great deal of work left to be done. And so if we are to really think about immunotherapy as being part of the foundation of a cure for cancer, then I think we have to be able to understand and address mechanisms of resistance, and that would increase response to checkpoint blockade, because soon the single largest population of cancer patients will be those whose cancer was either resistant or relapsed after checkpoint blockade, clearly underscoring the large unmet need. So as a field, we're clearly pursuing a wide variety of mechanisms to try and build on the success of checkpoint blockade. And this is really a broad net that's been cast and includes both, I don't know if you can see my, I don't know if you can see my pointer, but includes both therapeutic approaches to increase the inflammation in the tumor microenvironment, as well as to enhance the function of effector cells like NK cells and CD8 T cells in the microenvironment. Vaccination strategies obviously are important at increasing antigen delivery that might be important to elicit the activation of tumor-specific T cells in the draining lymph node, as well as a variety of approaches, cytokine-based approaches and others, that address the issue of trying to expand and improve the survival of populations of T cells that might have therapeutic effect. Clearly, the tumor microenvironment contains a range of mechanisms that can provide a strategy for the tumor to evade the effective immune response. And it's an interesting area of potential growth is the idea of being able to simultaneously invoke the immune response to cancer whilst simultaneously targeting intracellular tumor cell intrinsic pathways that tumor cells depend on for their growth and survival. So in many ways, this represents a spread bet that I think illustrates just how challenging the prospect is to try and identify the specific area of biology and the targets within that could be effectively combined with checkpoint blockade. And underscores what I think is sort of one of the truisms of drug development, which is that finding the right target is clearly the hardest part, has become the hardest part of drug development. So if one was to survey the most common reasons for failure, the mechanisms of failure across the spectrum of the drug development process over the last 20 or 30 years, it's clear that that's changing. In the 1990s, one of the largest reasons that a drug development campaign would fail would be because the PK properties, the pharmacokinetic properties of the drug, were just not simply good enough to deliver the right concentration of the drug to the right compartment to achieve the desired effect, the kind of proverbial brick dust floating around in a vial. That led to the incorporation of pharmacokinetic evaluation of molecules far earlier in the drug discovery process, which improved or was able to address many of the PK reasons for failure of drug development in a much earlier stage. In the 2000s, it became apparent that although we could now be much more effective at delivering compounds to the right compartment and the right concentrations, that many of those compounds were too toxic to be able to be developed successfully. That in turn led to the development of a series of increasingly sophisticated safety assessments that were also baked into very early stages of drug development that have now become much more successful at weeding out molecules that have an undesirable effect off and cause safety effects or toxicity. So this has really led to the kind of now this stubborn problem, which has been there all along, which is that the most common mechanism for failure in drug development is that the target's wrong. So our colleagues in medicinal chemistry and in protein sciences have become extremely good at developing compounds, molecules, and recombinant proteins that are that do just what they are intended. But as biologists, we've struggled to identify the right targets against which to make those drugs. So if we're to consider the development of new immunotherapeutics through the lens of how difficult that's going to be, what can we learn of recent history that might tell us whether this is whether immunotherapies are going to be more or easier or more difficult to develop to identify therapeutic targets in? So if I think about my career as an oncologist, I think there have been two broad waves of therapeutic discovery that have really changed how cancer is treated. One of them is obviously targeted therapy against signaling pathways that tumor cells depend on, and maybe the exemplar there that started off that revolution was imatinib, which obviously targets BCR-ABL. The second one, as I've already talked about, is the development of immunotherapies, and there the starting gun was really fired with the approval of ipilimumab targeting CTLA-4. And if one was to consider how many new targets, not how many drugs, but how many new targets were discovered or were the subject of successful drug approvals in the 10 years following the development of imatinib, the answer is that it's about 19 targets, depending on exactly how you define them. Targeted by more drugs than that, but 19 new biological targets. In contrast, in the decade following the approval of ipilimumab, there have been just 4 new immunotherapy targets approved, and that includes both PD-1 and PD-L1, which suggests to me that the discovery of new targets that will build on the successes of existing immunotherapeutics is going to be at least as difficult and probably much harder than therapeutic discovery in oncology to date. And that should come as no surprise. I think if you were to ask anybody who's been involved in the development efforts around vaccines for HIV or TB or malaria, I think they would probably agree that persuading the immune system to do what you want it to do therapeutically is far from a straightforward endeavor. So it's also true that even trying to identify new drugs that work synergistically with effective immunotherapeutics like PD-1 checkpoint blockade, even that task is not straightforward. My colleague Emmett Schmidt in the clinical organization at Merck Research Labs recently published a study which is a very nice meta-analysis of about 98 trials that encompass those where the monotherapy activity of PD-1 or PD-L1 blockade could be, and a combination could be assessed. So you could look at PD-1 plus X by themselves or together to determine whether the overall response rates achieved by combination therapy was better than that which you would have predicted from the monotherapy results for either independently in a way that was matched for stage and disease indication. And he applied the Bliss independence model, which basically asks whether that overall response rate is simply an additive effect of the 2 monotherapy response rates by themselves, or represents synergy, in which case you'd expect to see a greater overall response rate than would be predicted by the combination of the monotherapy response rates. And the data across these 98 trials suggests that basically any of the overall response rates that are seen in PD-1 or PD-L1 combination therapies is largely driven by simply addition of PD-1 monotherapy and the combination partner monotherapy, suggesting that we have not yet found ways to truly synergize between PD-1 checkpoint blockade and combination partners that are being used with it. So now that I've depressed the living daylights out of you by sort of laying out what I see as the challenges for immunotherapy development in the phase that we're in now, what's the reasonable response that one can take to try and expand the universe of targets that we could evaluate as to whether they indeed work well with checkpoint blockade, whether they're effective therapeutic targets? So much of what we know about immunotherapy we've learned from mouse models of syngeneic tumors, in which cell lines like B16 or CD28 or CD26 or MC38 are implanted into immunologically intact animals, where despite the presence of an intact immune system, they will grow inexorably. However, for many of these models, as I'm sure many of you are aware, the application of immunotherapy, including mouse avatars of checkpoint blockade or vaccination or adoptive T-cell therapy, under those circumstances, the tumors will shrink under selection pressure from the immune system. So it occurred to us several years ago that this may be an opportunity to try and use genetic selection screens to identify genetic perturbations of tumor cells that would confer a fitness advantage or a survival defect in the face of immune selection pressure. And the sort of scientific rationale for this is obviously founded in the types of genetic modifier screens that Drosophila geneticists have been using for half a century, where Drosophila with an intermediate eye size phenotype is genetically perturbed with E and U mutagenesis. And the identity of genes that actually enhance the phenotype by making the eye size smaller can be identified, as well as those genes that suppress the phenotype that actually revert eye size back to the wild type phenotype. And so in many ways, we can think of mouse syngeneic tumors in a similar vein, in which we can create an intermediate phenotype of a partial response to checkpoint blockade, for instance, in a mouse syngeneic tumor that has been perturbed genetically using CRISPR. And we can identify those perturbations that enhance the phenotype, i.e. that increase the response of that tumor to immunotherapy with checkpoint blockade, or alternatively that suppress the phenotype that cause outgrowth of the tumor. And these would obviously be genes that would be important to evaluate for immunotherapy targets, whereas these might represent resistance mechanisms. So we could obviously do this one by knocking out a single gene in each tumor, and you'd have one cohort of mice evaluate one gene, but that wouldn't get you very far in terms of the number of genes that you could evaluate feasibly. So what we really wanted to do was ask whether we could actually pool genes and actually ask in a cell intrinsic way whether loss or gain of function could be observed in a pooled setting. And the basis for this experiment was done by some very important experiments that were done by Natalie Collins when she was in the lab. Natalie is now a translational investigator in pediatric oncology at the Dana Farber. And she took B16 melanoma, the standard workhorse syngeneic tumor model, and transduced it so that it expressed either a relevant protein marked with a particular fluorophore or PD-L1, the cognate partner for PD-1, and those tumor cells could be distinguished based on the expression of different fluorochromes. She then implanted a mixed population of those cells, treated the animals with immunotherapy, in this case a GM-CSF secreting tumor vaccine developed by Glenn Dranoff many years ago, and asked whether there was a competitive advantage to the tumor cells that were expressing PD-L1. And indeed, that's exactly what she saw. So the input population of cells cultured in vitro remained at 50-50, and in an immunocompetent animal with no therapy, both the PD-L1 overexpressing and the wild type cells grew at equivalent rates. However, after in the setting of immune selection pressure elicited by an immune response generated by the vaccine, the tumors may be a little bit smaller, but those tumor cells that had overexpression of PD-L1 clearly won out. And that selection pressure was clearly was abrogated in the absence of an effective immune response. So if she depleted CD8 T cells, or did the experiment in a TCR alpha knockout animal that lacks conventional CD4s and CD8s, or indeed in a PD-1 knockout animal that lacked the receptor to which PDL would bind, those PD-L1 overexpressing tumors no longer had a competitive survival advantage. They grew at equivalent rates. The corollary was also true, in which when Natalie deleted PD-L1 using CRISPR-Cas9 and compared it to tumors that had wild type levels of PD-L1, she could detect a growth disadvantage. So here, this is shown as the relative proportions of cells with that were either wild type, shown in the red gate, or PD-L1 knockout in the blue gate, that were transduced with either control guide or PD-L1 in the blue gate. And as you can see, it's really only in the setting of immunocompetent animals treated with GVAX where loss of PD-L1 confers a growth disadvantage. So obviously, this allows one to compare the survival advantage or fitness cost of genetic deletion on a pairwise basis. But obviously, we were interested in using a broader library than that. And so Rob Mancuso took that initial observation and expanded it to a much larger scale, albeit subgenomic at that level, at that time, screen of pooled libraries of guide RNAs. Rob Mancuso was originally a graduate student and then a research scientist in my lab, and now has his own lab at MGH, where he's really doing terrific work to extend and really capitalize on this model that he built when he was in the lab. So in this setting, we take Cas9 expressing tumor cells, transduce them with a library of guide RNAs such that each guide RNA is represented a number of times, but any single cell only has one guide RNA in it. Treat with an immune selection pressure, and then establish the representation of those guide RNAs as a surrogate for the fitness cost or survival advantage conferred by loss of function of the indicated gene. And here we'd expect outgrowth of tumor cells deleted for a gene that might recapitulate a resistance mechanism, and similarly loss of the barcode representing a guide RNA that might represent an immunotherapy target because it increased the sensitivity of that tumor to immunotherapy. So when he did this with a library targeting about 2,000 genes, each of which targeted by 4 guide RNAs, these were the kinds of results that Rob found. So if we were to compare the distribution of guide RNAs in tumors that were grown in immunocompetent animals treated with immunotherapy, GVAX and PD-1 in this case, versus animals that lacked a functioning T-cell compartment and therefore could not apply an immune selection pressure, we see that the vast majority of the 10,000 or so guides that we use in this expression neither conferred a benefit nor a loss to the ability of tumor cells to grow in under immune selection pressure, and that's why you see this Gaussian curve at the top. However, a subset of those guides, indicated by the red bars in the lines below, clearly conferred a fitness cost on the tumor cell only in the setting of an intact immune system. In the presence, in the absence of immune selection pressure, the guides by and large cause no growth defects. So a couple of things were interesting about these results. Firstly, there are an awful lot of genes that, when deleted, cause selective sensitivity of tumor cells in the setting of an immune response. Perhaps even a larger number than we were imagining. Secondly, the types of genes that conferred sensitivity for the tumor tended to cluster into known pathways, suggesting that we were able to actually saturate a given biology or pathway at the level of genetic screening that we were doing. And thirdly, the striking result really was that there was an awful lot of biology that, as an immunologist, I probably wouldn't have been able to predict would confer sensitivity to a tumor cell. And in the next few slides, I just want to go through a couple of examples of this to sort of illustrate the kinds of genes the kinds of biology that we found. One of these being PTPN2, and another being ADAR. So PTPN2 is a phosphatase originally described as being expressed in T cells, but is actually much more widely expressed than that, which actually has a wide range of targets. But most importantly, and this is a little bit of a giveaway for the story here, is its role in dephosphorylating signaling intermediates downstream of interferon gamma. So in the absence of PTPN2, you get more pronounced interferon gamma signaling. So this is the result from the screen, where we see each of these 4 guides being markedly depleted under immune selection pressure. And when we made a tumor where all cells lacked PTPN2 in red, we can see that it's much more sensitive to immunotherapy here, PD-1 and GVAX, than our tumor cells transduced with a control guide. Part of the mechanism for this is that PTPN2 null tumors have a more pronounced immune infiltrate than do wild-type tumors. And the reason for that, Rob discovered, was because the enhanced interferon signaling that PTPN2 deficient cells experience results in increased expression of MHC class I and improved antigen presentation to infiltrating T cells, allowing them to much more readily identify tumor cells, become activated, and inflame the tumor. Tumor cells also, PTPN2 deficient tumor cells also have a tumor cell intrinsic defect in that they grow much, or they have much more pronounced growth defect when they experience interferon gamma than do wild type cells. So these show the results of competitive growth assays for wild type or PTPN2 deficient cells grown in a range of cytokine conditions, and for both mouse and human cells, loss of PTPN2 exacerbates the growth arrest and apoptosis induced by interferon. So what Rob really discovered here was that deletion of PTPN2 made tumors more sensitive to immunotherapy because it made them hyper-responsive to interferon gamma. It exaggerated growth arrest and apoptosis and increased antigen presentation to tumor-specific T cells. Another gene that Rob identified in this initial screen was a gene called adenosine deaminase. ADAR is a gene that normally catalyzes A to I editing in double-stranded RNA and prevents the sensing of invading viral RNAs and prevents them from activating and acts as a negative feedback loop to prevent exuberant activation of RNA sensors, including MDA5 and potentially RIG-I. That results in the elaboration of type 1 interferon and other interferon response genes and chemokines. The other RNA sensor that it prevents recognition of double-stranded RNA species by is protein kinase R, which is a serine threonine kinase that results on double-stranded RNA stimulation in translational arrest and apoptosis. Importantly, it's not just a negative feedback loop for viral RNAs, it also prevents sensing of SINEs, these short interspersed nuclear elements, which are the primordial leftovers of ancient viral infections that are scattered throughout the genome so densely they can actually form inverted repeats that, when transcribed, form double-stranded RNA species. And so they act as an important safeguard to prevent the untoward activation of the innate response by the genetic elements that represent a large fraction of our genome. In work that represented a collaboration from Jeff Ishizuka, who now runs his own lab at Yale, Jeff and Rob demonstrated that ADAR loss profoundly sensitized tumors to immunotherapy. And ADAR deficient tumors, shown in yellow or red here, grown in immunodeficient non-Hodgkin gamma mice, grow at more or less the same rate as wild type tumors, and in vitro they grow at exactly the same rate. In wild-type mice, however, that have an intact immune response, there's already a significant growth defect in B16 tumors that lack ADAR. And when those tumors are treated with PD-1 checkpoint blockade, B16 tumors are almost completely eradicated, which, for those of you who work with B16 tumors, will appreciate that this is quite a striking phenotype, because B16 is quite an immunotherapy-resistant tumor. Jeff and Rob went on to study the immune infiltrate in those tumors using single-cell RNA sequencing, and compared the immune infiltration control guide RNA transduced tumors, and could readily identify populations of immunoinhibitory populations like M2 macrophages and MDSC, as well as more favorable populations like monocytes and obviously CD8 T cells. In comparison, ADAR deficient tumor cells had a profoundly reshaped immune microenvironment with a significantly more prominent population of activated proliferating effector CD8 T cells. And now the monocyte population was much more prominent than these immunoinhibitory populations. And importantly, these populations of cells manifested a significant increase in type 1 interferon signatures. So how then was ADAR achieving this combination of inflammation and growth arrest, or reduction in size in the tumors in the setting of immunotherapy? Well, here Jeff and Rob used some really beautiful genetic epistasis by knocking out multiple genes in a single tumor cell, balanced by the appropriate number of control guide RNAs. So in a tumor cell that lacks ADAR by itself, as I've shown you before, it's significantly more sensitive to immunotherapy than tumor cells that have in which control guide RNAs have been introduced. if you delete in the same cell simultaneously ADAR and protein kinase R, the tumor cells are still sensitive to immunotherapy, suggesting that signaling through MDA5, presumably by enhancement of type 1 interferons and chemokines that recruit other immune effectors, is sufficient to mediate tumor cell enhanced sensitivity. Similarly, when you delete MDA5 or RIG-I for that matter, and leave PKR intact, the sensitivity of ADAR deficient cells is still preserved, suggesting that PKR and the resulting translational arrest or apoptosis by itself is also sufficient to mediate increased sensitivity of ADAR deficient cells to immunotherapy. However, it's only if you delete MDA5 and PKR that ADAR deficient cells really manifest. You can entirely reverse the increased sensitivity of ADAR deficient cells. An interesting mechanism for that is suggested by some work that we did in collaboration with Erez Lebanon at Bar Ilan University in Israel, which involved determining what sort of signs were those that were most subject to A to I editing by ADAR. So perhaps not surprisingly, edited signs occur in introns and 3' untranscribed regions. You'd sort of expect that because some transcription would need to occur in order to be able to form those double-stranded RNA structures. But really the surprise came when we found that the majority of, or that there was a great enrichment of ADAR-edited signs in interferon response genes, suggesting perhaps that one of the reasons we have as many signs in our genome as we do is because they might provide a very useful method to amplify the effect of interferon if the levels of those signs themselves are increased in expression on interferon exposure of the cell, and perhaps might contribute to the RNA sensing that a cell might experience in the setting of viral infection. But interesting work from Matthew Morrison's lab, led by Hugh Gannon, and by Rob McDonald's lab at Novartis, suggests that that mechanism may be important because they found that a subset of human cancer cell lines were exquisitely dependent on ADAR for their survival. And those subset of human cancer cell lines that at baseline were entirely dependent on ADAR were simultaneously those that had the highest expression, the highest spontaneous expression of interferon response genes, potentially pointing to the fact that those were genes that had a higher endogenous level of double-stranded RNA species to begin with, as one of the mechanisms that might explain that. The story behind this is quite complicated, so I won't go into this, but I think it's an interesting potential mechanistic link to explain why some tumors may be sensitive to ADAR inhibition. So what I've shown you is that ADAR deletion sensitizes tumors to immunotherapy by making them hyperresponsive to interferons through both tumor cell intrinsic mechanisms, by making them more sensitive to apoptosis mediated by PKR, and by tumor cell extrinsic mechanisms, resulting in the production of chemokines that recruit immune effector cells. Now, the observant amongst you might note that both PTPN2 and ADAR therefore function in analogous ways, by increasing the sensitivity to immunotherapy, by increasing the sensitivity of tumor cells themselves to making them hyperresponsive to interferons, although the biology downstream of that is obviously achieved via a different way. And data from our lab is not my lab is not the only one to make this observation. So, for instance, work from Kai Bucherfenig's lab demonstrated that in an in vitro screen to look for genes that sensitized tumor cell lines to cell death mediated by a TCR transgenic T cells. Amongst those cells, he certainly recovered PTPN2 as well, for instance, but also found genes that were the member of the PBAF complex, including PBRM1, AR2, and BRD7. And that the loss of PBRM1 clearly caused an increased sensitivity of those tumor cells to immunotherapy. And the mechanism, interestingly enough, appeared to be the fact that this chromatin modification complex, that's part of the switch-SNF biology, when deleted, made cells twitchier or more responsive to interferon gamma, again suggesting that the same mechanism, increased response to interferon, may be achieved through loss of PTBN2, loss of ADAR, or loss of PBRM1, although obviously the mechanisms of each of those is different. And importantly, Ellie Van Allen went on to show that this may not simply be an artifact of mouse models, but rather may represent an important mechanism in human disease as well, when he showed that patients with metastatic renal carcinoma who have truncating mutations in PBRM1 have a significantly increased response to checkpoint blockade, and that the RNA expression profiles of those PBRM truncated tumors were also enriched for genes that would suggest that they had enhanced chemokine responses. All of which raises an interesting question about the way in which immunotherapy induces an effective response against cancer. Our assumption, classically, is that a T cell engages with a tumor cell via interaction of TCR with a cognate peptide plus MHC complex. So it's basically, it's hand-to-hand combat. A T cell recognizes a neoepitope on a tumor cell and elaborates granzymes and perforin that result in the destruction of that tumor. And even cytokines like interferon are thought to be secreted in a directed fashion in a sort of hyperlocal way. But that is always, I find, a challenging model to explain how effective checkpoint blockade is, because any tumor cell may not necessarily be adjacent to a T cell. And obviously T cells certainly can migrate within the tumor cell body, but the math I've always found challenging about how a one-to-one killing effect of a T cell in a tumor cell can actually be affected in such a large lump of tumor relative to the relatively small number of T cells that are present, as shown here in this micrograph. And an explanation for this might be might come from some recent beautiful work from Ton Schumacher's lab, which demonstrated that actually T cells can exert an important field effect in tumors mediated via interferon gamma. He did this beautiful experiment where he took tumor cells that lacked an antigen that could be recognized, grew them in matrigel in the flank of an animal, and micro-injected a small population of tumor cells that could express an antigen that could be recognized. And the bulk population of cells that could not be recognized by T cells were transduced with an interferon reporter, allowing the stimulation of a Katushka fluorescence protein. He discovered that when he infused populations of T cells capable of recognizing the tumor cells in green, to his surprise, he found that the tumor cells that could not be recognized by the T cells showed a marked upregulation of the interferon reporter gene, suggesting and that the distance from those cells that were invisible to T cells could be as far as 800 micrometers, suggesting that there's quite a substantial field effect mediated by the interferon gamma elaborated by T cells that can itself have a profound growth inhibitory effect on tumors. So basically, what I've shown you so far is the work that's been done to try to identify targets within a tumor cell that could confer sensitivity to immunotherapy. But the other half of the equation is trying to identify genes expressed by T cells that could enhance the immunotherapeutic effect. The problem here, of course, is that the application of CRISPR genetic deletion is relatively straightforward in a tumor cell line, but it's really hard in T cells, because in order to allow them to incorporate either ribonucleoprotein or a viral vector, you really have to abuse them quite a lot, and the cells are no longer quite the same cells they were at the beginning of the experiment. So to address this, Marty Lafleur, who was a graduate student jointly supervised by Arlene Sharp and me, and who's now a research scientist in the Sharp lab, developed a really beautiful system to allow the introduction of guide RNAs into Cas9 expressing T cells. You're really getting the authentic home experience here of phones ringing, dogs barking. Sorry about that. The system that he developed allowed guide RNAs to be delivered to Cas9 expressing T cells in a way that doesn't perturb them. And he does this by making bone marrow chimeras in which guide RNAs are transduced into hematopoietic stem cells, and hematopoiesis is then reconstituted, including the development of, say, T cells, each of which contain the guide RNA and Cas9 in a way that doesn't allow those T cells to be subsequently manipulated. Those T cells can then be isolated and transferred to a new host and subject to a variety of experimental perturbations. Marty showed that he could achieve effective knockout in all of the kinds of immunological lineages that immunologists might be interested in, either at the level of protein expression or at the level of genetic deletion. And did some very careful barcoding experiments to identify that in any given animal, he could probably incorporate each animal could probably incorporate in their immune compartment something north of 500 guides, which a number that he subsequently has improved on in more recent experiments, suggesting that perhaps, although this may not be ready for, say, genome scale screens, certainly a very large chunk of the genome could be feasibly screened in cells in the immune system. So the first experiment that Marty was interested in investigating was whether he could identify genes that, when perturbed, would change the fate of exhausted T cells. As many of you know, I'm sure, exhausted T cells represent a state of T cell dysfunction that's seen under the settings of chronic viral infection and probably also cancer. Those cells have a range of functional defects, and really one of the cardinal ones of those is that they lose the ability to persist in the absence of antigen. Exhausted T cells can have heterogeneous fates and exist both in progenitor populations that have some residual proliferative capacity and terminally differentiated exhausted cells, which can't proliferate but are certainly very effective at being cytolytic effectors. Marty screened using this system a small library of about 110 guide RNAs targeting a handful of genes in the following gene categories. And he did this in a TCR transgenic background that allowed him to investigate the function of these genes in perpetuating the survival of exhausted T cells in the chronic LCMV model of T cell exhaustion, which is really the gold standard. So he generated bone marrow chimeras, isolated pools of transduced TCR transgenic naive CD8 T cells that were deleted for each of these 20 or 30 genes, transferred them now to a new host and subjected that host to a chronic viral infection that causes T cell exhaustion. And he simply counted up who were the winners and who were the losers amongst those genetically perturbed exhausted cells. The losers were kind of those that you might expect them to be. So T cells that were deleted for T cell signaling members like ZAP70 were clearly depleted from that pool, as were cells deleted for co-stimulatory molecules or other metabolic genes. But the genes that were enriched proved to be quite interesting. Deletion of the VEGF receptor or PD-1, a very useful positive control, or the adenosine receptor promoted the survival of exhausted T cells in the setting of chronic viral infection, as did our old friend PTPN2. Yeah, that's the same gene that I showed you as before as causing selective growth disadvantage to tumors in the setting of an immune attack. Marty confirmed indeed that loss of PTPN2 in a competitive mixing assay where a 50-50 mix of TCR transgenic wild type or PTPN2 deleted cells allowed the growth advantage of PTPN2 deficient cells to be clearly seen at multiple stages following chronic infection. Now, as I mentioned before, all exhausted cells are not exactly the same. Progenitor cells, which are truly exhausted, do undergo some renewal, some homeostatic proliferation, and are capable of differentiating into terminally differentiated cells. And these two populations of progenitor and terminally differentiated cells can be distinguished using surface markers. So Marty wanted to understand whether loss of PTPN2 caused a selective outgrowth of a particular subset of exhausted T cells, and indeed saw that in his experiments, what he was seeing was that PTPN2 caused a skewing or outgrowth of the terminally differentiated population of cells. shown here in terms of the absolute number of terminally differentiated cells or progenitor cells, you can see that the large growth survival advantage of exhausted cells was caused by an increased abundance of these terminally differentiated cells. Importantly, though, this wasn't at the expense of progenitor cells, which clearly existed at the same numbers. He demonstrated that indeed, as you might expect, PTPN2 deficient cells are hyperresponsive to interferon. They show when he removed them from an animal in the middle of a chronic infection, these exhausted cells showed a heightened ability to upregulate phospho-STAT1, an indicator of the fact that they're seeing more interferon signaling in the absence of PTPN2, as you might expect. And he showed that interferon signaling was necessary for this expansion of terminally differentiated cells. Using interferon blocking antibodies, he showed that the skewing that's present when animals are treated with a control antibody is entirely abrogated by the use of interferon blocking antibodies, suggesting that interferon signaling is necessary for this enhancement of skewing of terminally differentiated exhausted cells in the setting of PTPN2. In some careful work, which I don't have time to go into, he did lineage tracing of the cells and showed that PTPN2 null cells had a very complex phenotype in the exhausted compartment. Progenitor exhausted cells underwent increased proliferation to maintain their numbers and also fed into the terminally differentiated compartment at increased levels, and PTPN2 null terminally differentiated cells, surprisingly, were able to continually proliferate as well, accounting for their increased numbers. Now that's in the setting of chronic viral infection, but what about in the setting of cancer? So Brian Miller and Deb Sen in the lab, Brian who's now in Arlene's lab, and Deb, I'm delighted to say, will also be opening her own lab at MGH, have previously demonstrated in another paper from last year that tumor cells really convincingly, or tumor infiltrating T cells really convincingly demonstrate the hallmarks of exhaustion, including the presence of these terminally differentiated and progenitor populations. Marty confirmed that in the setting of syngeneic tumor models, PTPN2 deficient cells clearly had a survival advantage. And once again, we're seeing the skewing of those exhausted cells towards a terminally differentiated phenotype. He then went on to ask whether transfer of either wild type or PTPN2 deficient cells to a mouse bearing a tumor with a nominal antigen improved in equivalent or different levels of growth of control of tumor growth. And as you can see here in these B16 ova tumors, transfer of relative to transfer of control guide transduced T cells, OT1 T cells, PTPN2 knockout T cells improved the control of tumors significantly. And as you might expect, even in the absence of expression of an exogenous antigen like ovalbumin, B16 responded much to PD-1 point blockade in the immune system than did control cells. So what Marty was able to show was that he's developed a system that allows now the iterative screening of genes in T cells that might enhance their ability to confer anti-tumor effect. And that PTPN2 is the exemplar of that. In adoptively transferred T cells improved tumor control. And the complex dynamics of immune cells, of exhausted cell populations that is elicited by the loss of PTPN2 suggests that it is likely that exhausted cells, as we knew before, are still able to confer a significant degree of tumor control, but that it's probably both contributions both of the progenitor and of the terminally differentiated compartment that is important for their ability to control tumors. And PTPN2, of course, is a very interesting exemplar. I'm not sure how many others are like it, of a single target that is expressed in tumor cells and T cells with different but therapeutically useful effects in both. So largely the importance of this observation of Marty's system for identifying genes that can be used to modulate T cells is important for many reasons. Obviously, for the identification of immunotherapeutic targets that we can use potentially with checkpoint blockades to enhance its effect. But I think it has a role. I think these types of genetic studies that I think are enabled by Marty's platform, I think has a role beyond that in identifying the types of genetic modifications that we might want to make in adoptively transferred cells given as part of, say, a CAR T-cell therapeutic regimen. So I know that Carl June gave a talk on this seminar series relatively recently on this beautiful and important study that that he published relatively recently that demonstrated that it's entirely feasible now to genetically engineer CAR T cells, and that the introduction by ribonucleoproteins of guides targeting either the alpha or beta chain of the T cell receptor can delete the endogenous T cell and allow the more unopposed expression of an exogenous T cell, in this case the NY-ESO1 TCR. And he also included the deletion of PD-1 in order to determine whether that was feasible, obviously with the thought that if checkpoint blockade works by blocking PD-1, then perhaps genetic deletion of PD-1 might also enhance the function of cells. This is a very small study, and obviously a more detailed analysis and more patients is going to really be required to substantiate this finding. But I did think it was very interesting, and I think you commented on this as well, that loss of PD-1, that the cell populations that lacked PD-1 seem to have the worst degree of survival of all of those various permutations and modifications that he made. And I think this would probably be, would have been predicted by work from John Mary's lab and others that points to the important function of PD-1 to prevent the overactivation of T cells and their subsequent demise through activation-induced cell death. All this is to say that the identity of the genes that you would want to perturb in a T cell to improve the function of CAR T cells may not be straightforward to determine. And the genetic deletion of PD-1 may be a very different kind, may achieve a very different kind of biology than the temporary blockade of PD-1 with a monoclonal antibody. And tools to be able to actually do screens in primary T cells in a way that's enabled by Marty or by other platforms that I know are also being developed, I think will be an important part of the next phase of CAR T cell development, which I think is really going to be more reminiscent of synthetic biology than of sort of conventional adoptive T cell therapy. So in the last few minutes, I just wanted to share a couple of thoughts that I think have occurred to me recurrently over the last year or so since I've migrated across Longwood Avenue from the Dana Farber to Merck Research Laboratory for what it's worth. So I think it's pretty evident from a great deal of work done by a large number of labs that there's an enormous amount of biology out there that could potentially be targeted for the development of new immunotherapies. Clearly, I've talked about just one type of immune cell, the T cell, and the tumor cell itself, but clearly there are very complex populations, both of immune cells and of stromal cells that exist in a very complex ecosystem. And that opens the door to a very wide variety of biologies, both tumor cell intrinsically and in the tumor microenvironment, that could be usefully targeted to augment checkpoint blockade. However, identifying which of those mechanisms tumor cells depend on to evade the immune system, I think is going to be very difficult. We have grown used to identifying tumor cell dependencies, like KRAS, for instance, in an unequivocal way, because you can establish that through a variety of in vitro assays long before you ever get to try to inhibit KRAS in the clinic. Here, I think we have a much more complex system with many more moving parts, and the unequivocal identification of targets we know tumor cells depend on, I think, is going to be challenging. One area in particular that I think is particularly interesting to think about is to try and identify the intersection between the enormous amount of really important work that's been done understanding tumor cell intrinsic dependencies and trying to understand which of those also represent vulnerabilities for a tumor cell that is experiencing immune attack. The corollary of that first slide then is obviously that I would be the first to admit that I'm not smart enough to guess which are most important for therapeutic development. But I think we are in an era in science where systematic perturbation of biological systems offers an opportunity where maybe the most important biology can float to the surface. I've shown you some approaches with genetic perturbation, but I think the developments in chemical biology similarly offer a systematic approach to really try to understand nodes of vulnerability in a variety of pathways, like in interferon sensing or in innate signaling, that could render a tumor cell or a tumor more susceptible to the effects of immunotherapy. The corollary of that, of course, is that if we are able to identify those targets, say through in vitro screening or even in vivo screening, that it places a much higher burden on the predictive ability of model systems to tell us whether the targets that we maybe didn't know about before we did a screen of some sort are actually likely to be manifest in patients, and actually that the model system is an accurate avatar of the tumor immune interaction in people. And I think tumor explants and histocultures, tumor organoids that have intact immune systems that have intact immune elements, I should say, I think are really important additions to the armamentarium of model systems that can complement syngeneic tumors, which, as somebody much smarter than me once pointed out, are usually wrong, but sometimes can be useful. And finally, I think this may be stating bleeding obvious, but the most compelling experiment about where we should be going therapeutically, I think, is the human experiment. We obviously were heavily influenced in our selection of PTPN2 and ADAR as targets that might be interesting to pursue, based on the human germline genetics that pointed us in those directions. But I think the expanding clinical experience with checkpoint blockade offers a significant opportunity to really start to understand what the molecular features are of tumors that are more or less sensitive to checkpoint blockade. And I think even though Ellie's example is in just 35 patients or so, I think it's already become apparent that those types of analyses can point to important areas of biology for further study. And of course, understanding the sorts of state change that we're able to achieve using immunotherapeutic interventions in patients rather than in model systems, I think will be important for us to understand if we're actually doing what we think we're doing in patients treated with immunotherapy. So I will stop there and give my grateful thanks to the large number of Haining Lab alumni, a subset of whom are listed on the left and who are featured in that photo from pre-social distancing times. I've mentioned a few of them who played particularly instrumental roles in the data that I presented today, but really I think one of the things I miss most about my lab is the esprit de corps and synergy between lab members. I also particularly want to thank my colleagues at the Broad Institute, particularly Todd Golub, who's unswerving supportive of these types of systematic approaches to discovery. I think have always served as an inspiration for many of us, and I think led to the development and enabled many of the experiments that I've shown you today, as well as John Dench, who is, he's John Dench. What can I say? Arlene Sharp remains a terrific collaborator, and I'm lucky to have had collaborators like Matthew and Erez to help with the ADAR project. So David, I'll stop there, and in the last couple of minutes be happy to take any questions.
Well, thank you, Nick, for just an exceptional talk, and you handled the interruptions with your typical aplomb. There are many questions, and we will forward them to you, so you'll have a chance to respond to people if you have any inkling to do so. But I'll ask a couple. The first is somewhat of a challenging question. That is, you started by saying our major issue with drug development is that we don't do a good job of picking the right target. And as evidence, you showed 25,000 patients and 100 trials that demonstrated that we're not good at actually achieving synergy in patients. But I would guess that many of those trials were based on preclinical data using the B16 or A20 model that showed some extent of synergy. And then you spent the next 30 minutes explaining to us how you've identified new targets that can potentially synergize in those models. So where's the disconnect?
You accurately identified, Dave, what keeps me up at night. And I think there's no denying that syngeneic tumors offer a very poor avatar of human disease, in the sense that a B16 melanoma is really not the same as melanoma that occurs in patients. Having said that, I actually do think that mouse models can be extremely effective at understanding immunological mechanisms. So if we understand at a mechanistic basis what the objective is that we're trying to do, say we want to test the hypothesis that increasing interferon sensitivity in a tumor is a viable therapeutic approach, let's just say. I think mouse models do a particularly good, I think are a perfectly reasonable way of actually trying to prioritize the types of nodes, the types of regulatory points that could be usefully targeted, and to try to triage those. Then, of course, you have to ask the question: is it reasonable to think that enhancing interferon sensitivity in a patient would be useful? And there, I think work from Ellie or from Tony Rebus demonstrating the presence of STAT mutations in patients whose tumors have outgrown on tumors on checkpoint blockade becomes really, really important. So this is a very long way of saying, David, that I think that if you approach this from a basis of trying to identify how to manipulate an immunobiology in a tumor and combine that with the analysis of patient data to substantiate whether that mechanism is valid, I think you may be able to offset some of the limitations of mouse models.
Well, that's certainly the best answer I've heard to that question. So could you talk a little bit about your expectations for feasibility of targeting PTPN2, either pharmacologically or otherwise, and whether you would anticipate autoimmunity?
So, as a class, phosphatases have always been difficult, and so I think this would not be an easy target to go after. In terms of autoimmunity, I struggle with this a lot, because it's true that most of the immunotherapeutic approaches that have efficacy, PD-1 checkpoint blockade, CTLA-4 blockade, IL-2, bone marrow transplantation, are associated with significant immunological morbidity. And so a question that I think as a field we have to struggle with is whether it will really be possible to fully divorce efficacy of an immunotherapeutic approach from unwanted immunological consequences. I don't know the answer to that. So far, to me, it looks like it may be difficult to thread that needle, and we may have to be very skillful in the clinical development of effective immunotherapies, given that I suspect the effective ones are also going to be associated with immunopathology.
So a couple questions about ADAR. The first is whether it's been demonstrated that the catalytic activity is critical for its effect on tumor response, and the second, whether you think that the editing of introns is occurring in inappropriately retained introns, or is that after splicing of those introns?
Yeah. The first question is really good. The genetic data would suggest that some of the point mutations that occur in a syndrome called Aicardi-Goutieres syndrome, which some kindreds of whom have mutations in the ADAR gene, some of those mutations occur in the catalytic unit of ADAR. But it is complex, and I suspect whoever the questioner was knows indeed that ADAR can serve as a decoy receptor. Not decoy receptor isn't the right word. Can soft the RNA because it binds double-stranded RNA, and so simply actually subtracting double-stranded RNA species from the cytoplasm may be sufficient to reduce the amount of signaling that the RNA sensors MDA5 and PKR see. Reconstitution experiments with ADAR dead versions of with enzyme dead versions of ADAR are very difficult to do, but are clearly the right way to be able to quickly determine whether enzymatic inhibition will be sufficient for a sort of therapeutic approach. In terms of the RNA splicing, it's a great question. I actually don't know the answer to that. I think others who have a much deeper understanding of RNA biology would probably be better qualified than I am to the life cycle of trying to identify when that double-stranded RNA species caused by inverted repeats from signs is likely to form.
All right, well, I will stop it there. We're 5 minutes past the hour. We still have well over 500 participants online, and I think that bespeaks the enthusiasm at your talk. We have an extraordinary list of speakers next week. We'll start with Dana Farber from the Memorial Sloan Kettering on Monday, and then Scott Armstrong on Tuesday, Marcus Munchen on Thursday, and Judy Lieberman on Friday. I'd like to thank all the attendees and hope everyone is well. Again, we look forward to seeing you on Monday at noon Eastern Standard Time. Thanks again, Dr. Heenan.
Thank you, Dave. Thank you, Nelly, and stay safe, everybody.